So, how the acetyl CoA is exported from the mitochondria? The transport of acetyl CoA from the mitochondrial matrix to the cytosol heavily relies on the so-called citrate malate purate shuttle. In this context, the mitochondrial acetyl CoA is condensed with oxaloacetate by citrate synthesis, the first enzyme of the TC cycle generating citrate and free CoA. Citrate can be exported to the cytosol through SLC25A1, followed by the regeneration of oxaloacetate and acetyl CoA upon the ATP and CoA-dependent reaction catalyzed by ACLY. Cytosolic oxaloacetate is a substrate of the malate dehydrogenase 1, MDH1, catalyzing the NDH-dependent synthesis of malate, which can be transported back to the mitochondrial matrix via SLC25A10 or SLC25A11. Alternatively, malate enzyme 1, NDP-dependent cytosolic ME1, can convert malate into purate, which can re-enter the mitochondrial matrix via the MPC. Acetyl CoA is also exported from mitochondria via the kinetin shuttle. Thus, the citrate malate purate and the kinetin shuttle may ensure the continuous transfer of the acetyl CoA from mitochondria to the cytosol. Finally, acetyl CoA might be non-specifically released from the mitochondrial matrix upon transient openings of the so-called permeability transition pore complex, that is a multi-component channel built across the inner and outer mitochondrial membranes permitting to solutes that below 1.5 kD and involved in the regulation of cell death. Compartmentalized regulation of protein acetylation. The compartmentalization of acetyl CoA pores has a major impact on protein acetylation, be it non-enzymatic or be it catalyzed by ANTs or KATs. Specific acid transferase and deacetylase are also subject to subcellular compartmentalization. This applies, for instance, to EP300 as well as to the deacetylase 31, both of which shuttle between the nucleus and the cytosol. Another deacetylase of the family 33 localized for the most part to the mitochondrial matrix, and expression levels have been correlated with different patterns of mitochondrial protein acetylation, both in baseline conditions and in the course of chronic caloric restriction. So, cellular levels of acetyl CoA often varies. The abundance of acetyl CoA fluctuates in response to both intracellular and extracellular cues. Unfortunately, however, acetyl CoA is generally quantified in whole cell or whole organ extract, and very few studies have measured change in acetyl CoA concentrations at the subcellular level. So, how the relationship between the acetyl CoA and gene expression? In multiple cell types, histone acetylation is highly sensitive to the availability of the acetyl CoA. Acetyl CoA is indeed the obligate cofactor for histone acid transferase, meaning that the abundance of the nucleocytosolic pore may have a direct impact on the enzymatic activity of HATs, especially those with a relatively high KD for acetyl CoA. Thus, drops in nucleocytosolic acetyl CoA levels below the KD of specific HAT may directly reduce their activity. Moreover, several HATs, including GC5, are subject to product inhibition by free CoA. This indicates that the acetyl CoA and CoA ratio may be the relevant regulator of the enzymatic activity of HATs rather than absolute levels of acetyl CoA. Irrespective of the analyzing mechanisms, this establish a direct link between the nucleocytosolic abundance of acetyl CoA and the epigenetic control of gene expression. Indeed, high levels of histone acetylation are required not only to support global transcription, but also for the preferential transactivation of genes involved in cell growth and replication, glycolysis, and resistance to oxidative stress. Fluctuations in metabolites other than acetyl CoA also affect histone acetylation, hence impacting transcription both quantitatively and qualitatively. Besides feeding the TCA, the synthesis of some metabolites like hydroxybutyric inhibits HDAC1, HDAC2, and HDAC3. Along similar lines, L-catenin and sphingosine 1-phosphate favor histone acetylation by operating as HDAC1 and HDAC2 inhibitors. Conversely, increases in the ND and NDH ratio promote histone deacetylation by stimulating the activity of stertins. Also, variations in intracellular pH have a major impact on histone acetylation. Intracellular acidification promotes the activity of histone deacetylase as well as the export of proteinate acetate via monocarboxylate transporter as a pH buffer mechanism, whereas intracellular alkylation augments global histone acetylation. Of note, the chromatin of mammalian cells contains at least 10 potential acetylation sites, meaning that massive histone acetylation may cause a sizable depletion of nucleocytosolic acetyl CoA pole, hence impacting cellular metabolism. Conversely, the acetyl residues stored in histones may be mobilized to generate acetyl as an energy source. All these examples underscore the intricate links between metabolism and histone acetylation levels, which can influence gene expression both quantitatively and qualitatively. Fluctuations in the nucleocytosolic pole of acetyl CoA also modulate gene expression by altering the acetylation state of transcription factors. Thus, acetate stimulates aerosol poiesis in multiple mouse models of anemia by activating a signal pathway that involves the conversion of acetate into acetyl CoA by ACSA2. Other prominent transcription factors that are regulated by CBP or EP300 dependent acetylation as well as by SIRT1 or HDAC1 mediated deacetylation are Focal box O proteins and tumor protein P15-3. So, CoA and the cellular anabolism versus catabolism. Increase the nucleocytosolic acetyl CoA levels shift cellular metabolism toward anabolic reactions as they shut off catabolic circuitries. This effort is not limited to basic biochemical circuitries, but involves complex cellular programs. Thus, acetyl CoA levels affect the propensity of the cell to grow, progress along the cycle, mount autophagic responses to stress, and scramble to RCD. In addition, the abundance of acetyl CoA in defined cell types influences the metabolic relationship between different organs as well as behavioral cues such as appetite control. So, acetyl CoA regulates several cellular processes as it controls a balance between the anabolic and catabolic reactions, both as a central metabolic intermediate and as a signal transducer. Perhaps only ATP and ADH share with acetyl CoA such a key position in cell biology. Indeed, acetyl CoA not only is a substrate intermediate or product of vital bioenergetic circuitries, but also regulates several enzymes and influences the activity of a wide panel of processes, including the gene expression, via acetylation or deacetylation reactions. Okay, that's all. Thank you.